Okay, next question is question 3. This is going to be a shorter one. This one's only worth 4 marks. This one says, fireflies emit light when the enzyme luciferase catalyzes a reaction in which its substrate, D-luciferin, reacts to form oxyluciferin and other products seen in figure 1. In order to determine the optimal temperature of this enzyme, scientists added ATP to a solution containing D-luciferin, luciferase, and other substances needed for the reaction. They then measured the amount of light emitted during the first 3 seconds of the reaction when it was carried out at different temperatures. Okay. So again, like I usually say, the AP exam will throw new enzymes and stuff at you. This question is a general question about enzymes. They're giving you a specific example that you haven't heard of, which is luciferase. They give you the reaction, which makes it easy. So it's saying that luciferase is our enzyme. Again, an enzyme is not a reactant or a product, hence why it's above the arrow. It's acting on the reaction. It's not a product or a reactant. D-luciferin is going to react with O2 and ATP. Luciferase is going to catalyze it. You're going to make oxyluciferin, CO2, AMP, and then finally light. You need to understand the parameters of this reaction. So what they did was they tried these. They were trying to determine the optimal temperature. So this is really important because this is the goal of the experiment. They wanted to determine the optimal temperature that this enzyme works at. So what they did is they did several different reactions at different temperatures, right? If you're trying to see the effective temperature, you're going to want to do different temperatures for this reaction. You need a way to quantify that. So you need a way to quantify how much of the reaction is actually happening. In the case of luciferase, it's fairly easy because it's going to emit light. This one's easy. If this didn't emit light, you would probably have to do like the production of products. So like concentration of oxyluciferin or like the amount of CO2 released. In this case, light is being produced and that's how you know the reaction occurred. So they are measuring the amount of light being emitted at different temperatures to find which temperature is best for the enzyme. Okay, so that is the outline of the experiment. You don't need to know anything about luciferase. All right, so let's go into this one now. This one's pretty straightforward, I think. A says, describe a characteristic of the luciferase enzyme that allows it to catalyze the reaction. Okay, so I feel like students might get a little bit tripped up about this because they're asking you specifically about the luciferase enzyme. Now, they don't need you to say anything super specific about what the actual enzyme does. It's asking you pretty much in general. What is a characteristic about the luciferase enzyme, an enzyme in general, that allows it to catalyze a reaction? What allows enzymes to catalyze a reaction is basically what this is saying. So there are a couple of different things that you can say, but the main thing that allows an enzyme to catalyze a reaction is its matching shape with its substrate, right? So you have, this is your enzyme. So let's say this is luciferase. It really does matter. And then this is your substrate, D-luciferin. They will fit perfectly together, and that's what allows the reaction to catalyze. So that way that D-luciferin doesn't just react with anything, it will only react when it's bound with its partner, which is luciferase. That is general for all enzymes. It's not specific to luciferase. So for this one, I would say it has an active site or a shape that binds with the substrate. Has active site. Or you could also say shape that binds with substrate. Okay, so that's just a general property of all enzymes that allow them to catalyze reactions. It's because they have a compatible shape. Okay, so I think that's all you would have to say for A. Yeah, they would have accepted active site or shape that can bind the substrates, or you could have also said that bring, reactants together. Okay, B is asking you to identify the dependent variable. Again, very straightforward question. Like I always say, remember what your dependent variables mean before going in. Independent is what you're changing. Dependent is going to be what you're measuring. So your dependent variable is your y-axis. In this case, the thing that we're changing is temperature, and the thing that we're measuring is amount of light that's being emitted. So in this case, our dependent variable is the amount of light. They don't give you a unit here. If they did give you a unit, you should put it in. But in this case, they're just saying that you're measuring the amount of light emitted. So then our dependent variable is amount of light emitted. Okay, so that one's pretty straightforward. And if they asked you your independent, it's temperature. That's the thing that you're changing. C says, state the null hypothesis for this experiment. So you should also know what a null hypothesis is and what, the alternative hypothesis is before going into this. The null hypothesis is always the same for every single experiment. So as long as you remember what a null hypothesis is, you can always get these questions right. There's always one of them on the AP exam. The null hypothesis, null typically means nothing. So the term null is like synonymous with the term like nothing or no. So the null hypothesis is a statement that says that there is no correlation between the two variables, okay? And so you either accept the null hypothesis or reject after you do the experiment. Null always, always, always means that there is no correlation or no effect. So you state that as the null, as like the blank hypothesis, you can kind of say, is that there is no effect. And you should be specific. Don't just put no effect. No effect on amount of, sorry, let me rephrase that one. So temperature has no effect. Oops. You do have to include the variables in this. So what's affecting what? Temp has no effect on amount of light emitted. Okay, so this is the null hypothesis, and you either accept or reject based on this. So if I accept the null hypothesis, I'm saying that there's no effect. If I reject the null hypothesis, I'm saying that there is an effect. But the null hypothesis is always stating that there is no effect. Okay, so this one should be a freebie for you guys as long as you remember that. Okay, the final one, D, says a student claims that as temperature increases, there will be an increase in the amount of light given off by the reaction in the first 3 seconds. Support the student's claim. Okay, so you're not saying whether this is true or false. You have to support this. Okay. So basically, why would an increase in temperature cause an increase in amount of light emitted? Again, we're looking at enzymes, and if you guys remember from when we looked at factors affecting enzymes, temperature is always a big one, right? So what does temperature do to enzymes? It's very general, not just for luciferase. Temperature in general is going to speed up the rate of reaction, right? So that is why an increase in temperature is going to increase the amount of light. The more temperature you add, the more this reaction is going to go, the more light you're going to get as your end product. They're asking you to support this. So why would that be the case? And we do learn about this as to, why temperature increase causes an increase in reactions. Usually it's because it increases the kinetic energy and it's going to increase the amount of collisions, right? So for this reaction to occur, the luciferase has to come into contact with the D-luciferin, right? They don't just find each other, they will bump into each other. The more that they bump into each other, the more the reaction is going to happen, okay? So what heating up a reaction does is it makes the components move faster, right? You're increasing kinetic energy. Basically, your enzyme and your substrate are going to be moving a lot faster. That increases their chances of bumping into each other, okay? So increasing temperature is going to increase the rate of collisions. And then as a result, if your substrate and your enzyme are interacting more, you're going to get more product, which in this case is light, okay? So increase in temperature increases frequency of collisions, or you can say interactions. So we have collisions resulting in increased, you can say, production, which in this case is light. Okay, so you had to justify that. There was another thing that you could have said. This one is like the most obvious one in my opinion. This is the one that we teach you guys in the curriculum, that increase in temperature is going to increase the rate of reaction by increasing, the frequency of those collisions through kinetic energy. Another option that you could have done, because again, this is a pretty general thing that they're asking you. So they would have also accepted that the higher temperature makes the active site better at binding. This is also something that is possible. Not the first one that typically would come to students' minds or my mind, but it is possible that, maybe the active site is not in its proper shape at certain temperatures, and then when it increases in temperature, that the active site becomes, the proper shape, and then you can increase that. So what that would look like, I'm not going to write it down, but you could say that the higher temperature results in a change in active site that helps the substrate bind better. Okay, either one of those would work. Okay, I just think this one's much easier. For the AP exam, if you're between answers, pick whatever one is the easiest for you to explain, or the most simple. That's going to give you like the best marks. Okay, so don't overthink these. Go with the simplest answer. This is what we tell you in the curriculum. Just go with this one. But that one, in terms of like changing the active site shape, also accurate. Okay, so that was question 3. That one was pretty straightforward.